I think it's safe to say I've known longer than anybody else in this room has. So there are a few who are close, but is that true? I think that's true. Herman was my mentor long ago, more years than I think either of us wants to recollect, and he's been my colleague here at MIT for the last 35 years, and above all, he's been my friend for all of that time. He came to MIT at the beginning of the Cancer Center. I think he actually came a little bit before, because I remember he was in one of our old buildings before he moved over to the Cancer Center. And before that, he was chair of microbiology at Washington University for a moderate number of years. And before that, he was professor of dermatology at Washington University, which may seem like an odd transition, but actually it's not quite as mysterious as it sounds. It was actually the dermatology connection that first, I guess, brought him to my attention, and a little bit later me to his attention, because he visited Yale University, where I had gone to medical school and where I was had done some clinical work, and I was then doing a residency in dermatology, and he had been invited by the Aaron Lerner, who is the quite distinguished professor of dermatology and also a very good biochemist, to give a talk to our group. And I remember dimly that it was out at Aaron's house in a very nice suburb of New Haven. And Herman talked, I don't actually remember what he talked about, but it must have been about his work in delayed hypersensitivity, because that was connected to dermatology. And I remember being very interested in it, even though I'm now not sure, I don't actually remember in detail. But I then looked up his papers, and I was thinking of doing a postdoc in basic science, with the idea at that time probably of returning to a medical department. But everybody said that you should spend some years doing basic science before you did that. So I was thinking about that, and I was particularly struck by Herman's work when I heard it and when I read about it, because he was applying scientific principles to a very important clinical problem. And in my experience, that was fairly unusual in those days, especially. And perhaps he'll say a little bit more about it, but he was essentially working on how it is that if you're exposed to poison ivy, what happens to make you allergic to it. And he approached it from a biochemical point of view, and I found that very interesting. So in due course, I ended up as a postdoc at Washington University, and I'd like now to just take a few minutes of his time I started to reflect when I was going to do this about what things were like in those days, which is the early 60s. And so let's start with what was the world like? Well, the Vietnam War was heating up. The civil rights movement was in full swing. And what else did I... Anyway, a few other things were happening. Oh yes, Kennedy was assassinated. And I remember that very distinctly because everybody remembers. There are two events in my lifetime that people remember. One was Pearl Harbor. Those of you who are old enough to remember, everybody remembers exactly what they were doing when they heard about the attack on Pearl Harbor. Everybody, again, those perhaps slightly younger, but still somewhat old, remember what they were doing when Kennedy was assassinated. And I was pipetting at the bench and when this news came, and of course it was very shocking. What was biology like in those days? Well, it was, of course, a few years, not that many years past Watson and Crick. The genetic code had just been unraveled. And quite important for what I was interested in, Sanger had shown by sequencing insulin not that many years earlier that proteins were actually defined molecules with a distinct sequence of amino acids, which had really not been clear until he determined the sequence. And by that time, ribonuclease had also been sequenced and possibly lysozyme, but it was in the very early days of our understanding of proteins. What was immunology like at that time? Well, people were still arguing instruction versus selection, although I think the arguments were petering out. We knew practically nothing about the structure of antibody molecules themselves, and but that began to happen during the time I was in the lab, and in fact, one of the participants in that in those events is right here in the audience, Julian Fleischman, who worked with Rodney Porter and doing groundbreaking work on unraveling the polypeptide structure of antibodies. But certainly nothing was known about their sequence, and everybody was contending with what was called the antibody problem, which is what concerned Herman also and a small group of other people who formed what was known as the Antibody Workshop that met once a year or so for, I don't know, 5, 6, 7 years to discuss what was considered at that time the central problem in the field, at least most people interested in antibodies. What determined the amazing specificity and diversity of antibody molecules? How is this chemically coded in the molecule? And there was lots of discussion about that. At that time, it had just been shown that the lymphocyte was actually involved in immune reactions. And when I was in medical school, just a few years before that, nobody had any idea what lymphocytes did. They were the most prevalent cell in the blood. Everybody could see them in great quantity, but nobody had a clue as to what they were doing. So by this time, it was shown that lymphocytes actually made antibodies, and plasma cells specifically made antibodies. So the lymphocyte was important, but B and T cells were yet on the horizon. So we were really abysmally ignorant, if you consider it relative to what we know today. What were the labs like? What was the department like at Washington University in those days? Well, the department was pretty small. Herman more or less assembled it from scratch. Went around the world, I think, seeking people to hire. The other people in the department were all young assistant professors. The labs were all small. I think Herman's lab at the time, as I recollect, had 3 postdocs, a couple of technicians, and that was about it. The other labs were even smaller with these young assistant professors. Everybody knew everybody, and there were a lot of interactions. The other thing that I remembered about those days were that everybody mouth pipetted, which is sort of those of you who are old enough will remember that. Anyway, that was so. My own project, which was to try to figure out why antibodies changed in their binding constant over a period of time after immunization, a property which somehow has now gotten the term of affinity maturation. I had a lot of fun with it, and I remember the feeling that I think many people starting out in full-time research have, of that it was so amazing to be paid for doing something that was so much fun. So it was a very fruitful time for me. And getting back to Herman, though, we had a lot of discussions, and at that time, and still today, we interact virtually on a daily basis. We discuss immunology, we discuss horticulture occasionally. The flow of information is still mainly from Herman to me, and we discuss the news sometimes. And I've also, the other thing that has really just amazed me in our interaction is that I find Herman essentially unchanged since I first knew him so many years ago. He's still got this intense focus on science. He still discusses it. He understands it. He hasn't lost any bit of concentration about it, and I find that both inspiring and amazing. And I think I'll stop here.
Keep going. You're doing fine. I'm enjoying it now.
No, he doesn't have anything.
Yeah, that's all right. It's fine. Okay, so it wasn't quite clear what I'm supposed to do here, but I don't want to bore you with reminiscences, but I will mention a couple of things because it is sort of pertinent what Lisa was saying about her first encountering me as a dermatologist. So how did I get to be a dermatologist, you might ask? Well, I was an MD. I did a residency in pathology at Columbia in the just around the end of World War II, and it was a wonderful department of biochemistry there, headed by a man named Hans Clark, who had assembled some great biochemist refugees from Hitler's Europe, and also had some wonderful graduate students, Conrad Bloch, David Shemin, Seymour Cohn, and then Boris Magasanik was one of them. It was a very brilliant collection of graduate students. I was well aware of that as a resident just doing pathology, but I sort of gravitated to the laboratory there of a man named Michael Heidelberg, whom none of you probably know except for Boris, Jean, Raj, who was an organic chemist who had gone into immunology sort of in a backhanded way because he had been at Rockefeller and asked by the head of the Rockefeller Institute at the time to help out Oswald Avery, who was working on not DNA then, but on the pneumococcus, from which he did later discover DNA, and trying to determine what was the major antigen in the pneumococcus. Pneumococcus was then what HIV virus is now. It was a major pathogen. It caused a lot of disease every year. In New York City, the hospitals where I was a resident would see floods of patients with pneumococcal pneumonia. With some strains of pneumococcus, the mortalities were high. It would be as high as 25%, some occasionally. And a large group was devoted to just studying the pneumococcus. And so Oswald Avery decided they really should try to find out about the major antigen in this organism. And Michael Heidelberg was enlisted to help him. And so they grew tons of a particular pneumococcus and started to isolate the major antigen, which was assumed to be a protein, like all other known antigens. And they were doing Keldol analysis to measure nitrogen determination to follow the isolation of the antigen using antibodies they got from serum of patients. Yesterday, the antibodies in those days were known just as an activity in serum. They were never purified. Some people didn't argue they were not even proteins because you could dilute a serum and still show antibody activity and find no protein by any of the known methods. So there's a debate going on to some extent. Anyway, as they purified this pneumococcus antigen more and more, the nitrogen content kept going down and down. So it was very puzzling until Avery suggested, hey, maybe it's not even a protein, maybe it's a carbohydrate, which indeed is what it turned out to be. But to prove it took a lot of heroic work because the methods then available were not at all simple. Heidelberg then spent the rest of his long life studying different pneumococcal polysaccharides and their structures. But one of the things that came out of that was that using a purified polysaccharide as an antigen, they could precipitate antibodies from serum. And in the precipitate, they could see there was really protein. And they could learn how to isolate the protein back in the precipitate and got back purified antibody. And that was really absolute clear demonstration about antibodies were proteins, and the protein structure was then started in the very primitive way, just learning about molecular weights and electrophoretic behavior and so on. Well, I went back to NYU, and here's what I'm building up to about the dermatology part. I became one of the first, luckily, one of the first appointees of what would now be called a Physician Scientist Award, given out by the NIH. Up until World War II, practically all research in the country was supported by private philanthropy, no NIH. The experience of World War II had such a big impact in convincing government officials about the importance of science for defense of the country, and not only support of physical and chemistry, but also support of biology, that they started these fellowships going, and I got one of them, and that was really enabled me to do escape from doing a lot of clinical work. But in any case, I was interested in working on antibodies, the only things that were known about the immune system at that time. And Fred Sanger, who is always one of my heroes, came through to give a seminar in a room that was probably half this size, with half the number of people, to talk about his work on insulin. He was then working out the way of doing sequencing of insulin to establish that proteins were made by amino acids linked together in alpha peptide bonds. At that time, still a matter of dispute about how proteins are constructed. And he talked about DNCB and dinitrochlorobenzene as a way of labeling the end groups on peptides, then dinitrofluorobenzene. He didn't succeed he made big headway with dinitrochlorobenzene, but with dinitrofluorobenzene, he really was able to work very effectively because it reacted with every amino acid alpha amino group, whereas dinitrochlorobenzene wasn't nearly as reactive. And so when he finished with that talk, which led eventually to the publication that established the amino acid sequence basis for protein structure, and his winning the Nobel Prize, I just thought I wanted to sequence an antibody, a purified antibody. So I thought, you want to use dinitrofluorobenzene. Well, you couldn't buy it then, you had to make it. So I synthesized it, according to recipes in the book, and I got a terrific rash. I became very sensitive to dinitrofluorobenzene. Well, that was kind of interesting because then I was able to do skin tests on myself with all kinds of dinitrobenzenes, and also on guinea pigs. And one thing led to another, but I led to some papers we were able to publish some papers on the structure of the nitrobenzenes and their relationship to delayed type hypersensitivity. At that time, because T cells weren't known, I was really studying a T cell reaction in my own skin and on guinea pig skin. And I published these papers, and then I was visited in my lab at NYU by a man named Barry Wood, whom probably none of you know, a very charismatic professor of medicine at Washington U. So he asked if I was interested in a job there. And the job was to be head of dermatology. Why? Barry Wood was one of the directors of the Rockefeller Foundation. And he persuaded them that there had to be some very decent basic research in skin disease within departments of medicine. And so they endowed a chair at Washington U for that purpose. And I was recruited for that job. So that's how I ended up in dermatology. And I was in it for a while, and then Arthur Kornberg when I went to Washington U, as often happens, I was supposed to have a laboratory all built and ready for me. It didn't get finished for 6 months. During that 6 months, Arthur Kornberg, who was head of microbiology at Washington U, gave me space in their labs. So I spent 6 months in that wonderful department, which was also very small, with a lot of assistant professors like Paul Berg and Dale Kaiser, who was Chris Kaiser's father, Dave Hogness, Bob Blayman. It was a terrific small group. It was a wonderful experience. And then Arthur was recruited to go to, Kornberg was recruited to go to Stanford. That chair became vacated, and I got recruited into that job. And that's how I ended up in microbiology. And had a fine time studying antibodies. But then, being a department chairman at a medical school, you can't realize what a special kind of academic job that is.
Political?
Huh? Very political. A lot of administrative things, not just the department, that was fine, but all kinds of hospital problems. And I got progressively unhappy about having to spend a lot of time away doing those things. And so after a while, I was in that job though for 13 years, but then was offered a job at Columbia and at MIT about the same time. And I think I ran into Gene Brown somewhere. And he knew about this. He said, don't make up your mind yet. Wait a while until you hear more about it. So in any event, they ended up came to MIT. I had enough of medical school for all that time. And happily, I made the right decision. And so when I got to MIT, again, I continued working on immunoglobulins because we knew a lot about the structure. Myeloma proteins were turned out to be made by myeloma tumors. And they were wonderful molecules for studying not only antibody structure, but antibody function, because it turns out if you did a right kind of screening, as I was able to do, I'll tell you how in a moment, you could find tumor proteins made by tumors that will react very specifically with a given antigen or a hapten, like the dinitrobenzenes I had been using as ligands for antibodies. And that work was continued. I won't tell you a lot about those details, but that work was continued here at MIT. And then after a couple of years, T cells had been discovered. And I thought we should start looking at T cells which presumably had structures on their surface like antibodies, but those receptors bound antigens were not known, nor were their ligands known. And so every postdoc coming to the lab, I asked them to isolate a clone of T cells. And a lot of T cell clones were made that way, and one of them grew very well. That's one we call 2C, and it's still being used in various ways now. But I think that maybe at this point, I should stop and see whether people have questions.
Herman, when you entered medical school, did you think you wanted to be a practicing physician?
I had no other thought in mind because the whole medical school faculty at NYU, where I was a student, were all practicing physicians, except for a few appointees in the preclinical departments, and they weren't MDs, they were all PhDs. So there was no possibility except to be a practitioner and to try to do a little bit of research on the side.
Did you actually practice medicine for a while?
Oh yes, I did. I did for a while. For about 5 years I had an office, saw patients, delivered babies. But I had a grant from the Standard, well, it doesn't exist anymore, the Standard Oil Company of New Jersey gave the medical school a $10,000 a year grant to support a lab and a faculty member to do research because of this contact skin dermatitis, this contact skin allergy problems we've been studying, delayed type hypersensitivity in the skin. So that's the job, that's the appointment I had. And I was supposed to practice medicine half-time in an office a few blocks away and spend a few hours a day in the lab on this. So I ended up by spending all day in the lab, getting to the office at 6 o'clock in the evening, and getting home at 10 or 11 at night, and having a very unhappy wife.
I think you were just at the intersection where it was becoming impossible to have a private practice and do research at the same time. Because I know my brother-in-law, who was much, much older than I, big brother-in-law, was a physician and definitely had private practice, and that sustained him. But he also had a research appointment at Mount Sinai in New York and did research on diabetes, SPRU. And it was still possible, but I think
it was possible
before your time. I think at your time it had already become possible.
What Boris is saying is that that whole combination of trying to do clinical practice and research was becoming progressively untenable as time went on. That's absolutely true, but for a long time, Boris, it's amazing how people in clinical departments of medical school were expected to be able to teach, to do a lot of research, and to be good clinicians. All three, triple threats, like in to use a football terminology. But you're right, it became untenable.
I found...
I'm sorry, just what were the years that you were doing, when you were doing those 5 years of practice?
1950 to 55, or 4
And
years.
that's when you were on...
I was at that time, let's see, 50, I spent a few months, almost about a year at Sloan Kettering. Went back to NYU
So that's
with a faculty appointment. Yeah, and had that grant.
So you definitely
50 to 55.
You definitely wanted to be an academician.
Oh yeah, I wanted to be an academic. Yeah, but you know, there's a whole tradition for amateurs doing research. It goes back a long time, hundreds of years. hadn't died yet. It was dying. We didn't know it. But amateurs did research. A lot of research was done by amateurs.
Well, when I was in the Department of Medicine at Yale, I thought there were amateurs doing research. I didn't realize it at the time, but when I went into Herman's department where there were professionals doing research, it was a big difference. I don't know if that's still true. It's probably not true anymore.
I think it's not true. I think there's recognition in medical schools now that there are clinicians, there are researchers.
So you had to make a decision then, didn't you? About whether or not you want to continue to practice medicine or do full-time research.
Well, I knew what I wanted to do, but the decision, in a sense, was made for me by Barry Woods offering me that chance to become full-time. Yeah.
Herman, was your offer to Columbia offer to go to medical school, or?
It was to be chairman of microbiology at Columbia, like what it was at Washington U.
So was that one of the reasons why you came to MIT?
Yes. Right. Yes. No, because at MIT, I found an entirely different environment for doing science. It was really wonderful. You know, it was uncontaminated by anything else. The teaching was focused, research was focused. It was, but I don't want to be too extreme about this because there are wonderful things go on in medical schools, but it's difficult. And I think there's now this separation into groups at the medical schools. To those.
The person who was really instrumental in bringing you here was Oswald Avery. Of course, his background is a similar one. He was an MD and went into full-time research.
Yes. But bringing up Salvo Luria, that's really what got me very interested in MIT. Because Salvo was at that, let's see, in around 1970. Salvo was beginning to plan the cancer center at MIT. And he wrote to me and said, you know, I'm writing this grant application to establish a cancer center, and I need some input about the immunology of cancer, tumor immunology. So we had a correspondence for a while about that. Then he asked me for references about all of that. And we were doing some work on myeloma tumors that was related to immune system causing rejection of myeloma tumors. And that led to his really promoting my coming. And I came here actually to MIT, but it was to be in the cancer center, but to be devoting about half my time to the HST program, the Harvard MIT program. But the nice thing about being at MIT was that there's a lot of flexibility. And you can sort of gravitate to the place where you want to be. So I gravitated to where I wanted to be. Yeah.
Herman, could you make some comments how writing grants and writing papers has changed over the years from your perspective?
Oh, indeed, yeah. Well, I know writing grants was relatively easy. In retrospect, it was really pretty easy. For one thing, the NIH at that time really had money, and if you had a halfway decent proposal, you were going to get funded. So it was only a question of trying to satisfy your own sense of your own standards that made you write a strong application so you'd get a good high score. But the funding was pretty straightforward, and it was available. And of course, the whole NIH support of research became so strong that the dependence on private philanthropy became less and less. Now, of course, one begins to see the reverse, where private philanthropy becomes increasingly important, and grants from companies become important. Whereas NIHs get money is harder and harder to get. I wanted to mention something about that, about private philanthropy and grants. I was on the Howard Hughes advisory board for about 10 years when they were giving out their support for Howard Hughes investigators. At that time, they were limited they limited themselves to giving support to Howard Hughes investigators in hospitals only, for very complicated reasons having to do with the Internal Revenue Service and taxing structures. But with a lot of pressure from many of us on the board, they began to also support research in non-hospitals and medical schools and then universities like MIT, so the Howard Hughes investigators here. But the Howard Hughes now has formed its own big new research establishment at General Farm, and I'm not very happy about that because I think it's at a time when that money could be used for supporting research elsewhere. When
Are they going to support any research elsewhere, or is it only going to be in their university?
Oh no, they're supporting research elsewhere too, but a lot of their resources are now have to go to that farm, that special one.
Herman, what was your early life like? I mean, how did you decide to make the decision to go to medical school and so forth?
Well, I decided, in high school, chemistry appealed to me and nothing else. It's a testimony, I guess, to how generally poor my high school education was, but for some reason, chemistry was appealing. And,
Where was that?
huh?
Where was that?
In New York, in the public school system. And then I thought I'd like to be a chemist or a chemical engineer, but being Jewish, my father persuaded me that there'd be very few job opportunities in chemical industry. I should go into medicine, because in medicine you're a sort of free agent. You do what you want to do practically. So I was persuaded to go into become a pre-med for that reason. But even in medical school, I was always really interested in science, particularly in chemistry. And I think that interest really was driven home by experience I had once between jobs, between being at Columbia and coming back to NYU. I took an extended vacation as a ship surgeon on a cargo ship going down to South America. And it's stopping at ports along the way to unload and load up things. And you get very busy during those times because a lot of accidents. I was very busy with that. But in between times, I had completely free time. And I'd taken along with me a copy of a monograph by Carl Landsteiner called The Specificity of Serological Reactions. A fantastic summary of what was then known about immunology. I was very interested in immunology from my exposure to Michael Heidelberg at Columbia. And that monograph, which I used to recommend to everybody, every student to read, is, I still have, is well worn, and no one ever look at it anymore. It's really not much interest to people. In fact, I dare say the immunologists here, the young, have they even heard of Carl Landsteiner? Raise your hand if you have. Yeah, no, if you, you're not allowed. How about the younger ones in the back? How many of you heard of Carl Landsteiner? How many of you heard of Michael Heidelberg? Amazing.
Well, if they take immunology, they hear about.
From you. Thank goodness.
But after I go.
Yeah. Yeah.
Herman, as you trace the development of immunology, would you say that chemistry played an important role in the early days?
You know, I had occasion, I wrote a textbook with Bernie Davis and some others, Phil Becko and others, microbiology and immunology. And I looked at it not so long ago, and it's sort of amazing to me. This is that book was written or started 30 years ago, how it's chemical immunology. That was what appealed to me, and that was a really good, what I considered hard science in chemistry and immunology then. The cellular immunology, I always thought was pretty soft stuff. And of course, it's been amazing to see how it's changed. I think now, between cell immunology and molecular immunology, there is a continuum. So it's become like the rest of biology. It's molecular cell biology. And that's been a very gratifying thing to see. But in the old days, it was primarily chemical immunology. And that was, there's some very good stuff being done. I mean, in fact, a lot more was known about how protein, small molecule interactions took place. was analyzed in greater depth than it would be nowadays. Nowadays, I think there's such a preoccupation with identifying the players with names and acronyms I can never keep track of. The ZAP-70 is one of them, lucky to remember. Just running around identifying who the players are without really worrying about how they interact with each other. Then we just worried about an antibody, a purified antibody, a small molecule like a dinitrobenzene, dinitrotoluene. It's changed dramatically. I would say for the better, too. But I would say for the younger people here, a big part, I think, if I look back about what made things sort of work to the extent that they did work for me, was being able to take advantage of what was in the environment. Take advantage of opportunities in the environment. You know? Not just so much planning, having a game plan, but responding to interesting possibilities.
And also doing something you like to do.
Yes, absolutely. Critical, essential. Yes.
When you say the environment, I mean, you're not just talking about what the results of an experiment were, but something else like skin rash?
Well, I'll give you two examples. Here are two examples. When Arthur Kornberg's group left to go to Stanford, they left behind some equipment they weren't interested in. One of them was free electrophoresis, which nobody wanted to use anymore. The other one was a split beam spectrophotometer. Split beam spectrophotometer turned out to be fantastic for me because with a split beam spectrophotometer, you were able to mix a hapten, like DNP lysine, with the serum that contained the myeloma protein. And if it bound, I knew there was a spectral shift of the hapten. If it was bound to an immunoglobulin. If it bound to albumin, which many things bind to, you get a different kind of spectral shift. So I learned with this double beam spectrophotometer, we were able to screen serum from patients with myeloma tumors about 1 or 2 at a minute, what would now be called a high throughput screen. And there was a guy around who had been collecting, named Kurt Gusland, who'd been collecting these serum samples from patients for years. He had a pretty good library. So he gave me these, I'd get 1 or 2 mils of serum from him. I could screen all these myeloma tumors, and we came up with a myeloma protein that bound DNP lysine. Well, that was a terrific opening into looking at how this antibody-like molecule, and indeed it was an antibody, but made by a tumor and not in response to any antigen, how it bound haptens. That opened up a lot of interesting work. The other example I'll give you is there was a colleague at Washington U named Sidney Velick. I don't know how many of you know Sidney Velick, one of the best protein chemists around. Very quiet, unassuming, and but terrific. Velick has started to use forced energy resonance transfer to look at the binding of pyridine nucleotides to enzymes. Looking at what's now called FRET. Many of you know that FRET. So I heard him talk about this once, and I talked to him about it afterwards because we realized that the dinitrobenzene haptens we were using with various antibodies had a spectral overlap with the tryptophan omission from the antibody. So he and I talked about the possibility of putting together my purified antibodies and haptens with his looking at the energy transfer system. We did an experiment together after a few months, and it was a spectacular experiment. You could do energy transfer and look at hapten binding with small amounts of antibody, microgram amounts, instead of the amounts we used to use for equilibrium analysis. And they were able to do really study serial bleedings from mice, from rabbits, and study affinity maturation and do all kinds of things by taking advantage of that FRET, which I would have only learned about from hearing Sid Velick chatter about it at a meeting.
Were you, during the Second World War, were you in service? Or?
No, I was supposed to go into service. I had had tuberculosis in my late teens, and I thought it wouldn't matter, but they wouldn't. I was rejected, and I must say I was very disappointed by that, and didn't serve in the Army as a result, but instead I had to do a lot more medicine.
When you said you had tuberculosis, it was when you were at medical school, because it was very common, I think, people doing postmortems
So it was very common.
becoming infected.
There was no treatment except for bed rest.
That was your case too?
Just before I got into medical school.
Or just before you?
Yeah.
So it was not due to,
No, it was not due to exposure in medical school.
it was very common, I thought.
It was common, yes. Well, I would say that in a medical school class, you did a tuberculin skin test, a delayed hypersensitivity test, look at tuberculin. I would say 3/4 of the class would have been tuberculin positive.
When I take the tuberculin test, they used to have to give it to me all the time. You know, you go to a different school, my arm was swelled.
With swells like that, mine too.
Because I had my grandfather died of TB, and then one of my aunts and one of my uncles died of TB, and we were all interacting that whole time.
It was if you did a tuberculin test now on this population here, let's say those under 50, I think you'd find very few would be tuberculin positive.
So it really has become much less
Yeah.
common.
Should we go to VA hospital?
All right.
Well, you've been satisfied here at MIT?
Yeah. Thank you, Gene. And Boris. Boris was chairman when I came. No, I've been made the right move for me.
So Herman, I don't want to belabor this point. When I asked my question, I really had two parts to it, and the second part was about papers. And I just want to say this as a compliment to you, because as a graduate student in the 1960s, your papers were the first papers that I followed, and I thought they were fabulous. Whatever you wrote was right, and you said this many microliters of this, or maybe it was no liters then. Everything worked. It was like a cookbook. And now the papers seem to have mistakes and sloppiness, and it's very disconcerting, because when my students can't follow it, it turns out it's really not their fault that the papers are not well edited. Do you find that also, and what do you think we can do about that?
It is true. I think people are under much more pressure to, first of all, publication. How many papers are published? I mentioned Fred Sanger before, one of my heroes. I don't think Fred Sanger published more than 50, 60 papers in a lifetime. But they were really great. I don't know how many Oswald. I think that there's a tendency now to publish a lot because there's a lot of pressure from the NIH for grant purposes, and there's just but I will say this about the papers, although there may be a certain amount of sloppiness and a lot of papers being written, the information content per paper now is much higher than it used to be. I think it's just because you can do so much more is expected in them. But the papers that you refer, maybe some of the papers you're referring to, came from my lab from when Lisa was a postdoc. She finished work and had left the lab to go to Porter's lab. We wrote slowly and published slowly, and I think she had 3 papers back to back in the Journal of Experimental Medicine, which were pretty, but they were published after she had left the lab, I think.
After I left Porter's lab, too.
So if they were edited carefully, it was a very slow process. But I didn't worry about being scooped.
The standard used to be that for writing a paper that somebody else should be able to pick it up and repeat exactly what you did.
Exactly.
And this is no longer the standard.
It's not my imagination.
No. And the other thing I was going to say, and I think it was one of the points I forgot, was a minor point, but this average paper in those days had 2 authors. The senior person and the person who did the work. That was the average paper. I mean, a paper with 3 or 4 authors was unusual.
Now you have 15 or 20.
Well, it's also a matter of style. Bob Burgeow, when he was here, when he was dean, before you or after you? After you.
I appointed Bob as...
You appointed Bob. So Birgeneau, who's a physicist, his attitude about authorship on papers was that anybody who had anything to do with could be on it. Some people are very stringent about how they look at, others are much less so. It's a lot of taste, a lot of variation in taste about that. But some of the large numbers of authors come because of these big consortia carrying out research. Yeah. Well.
What are some of the greatest, moments you remember while at MIT for you personally?
You can say that again.
Some of the, most satisfying or greatest moments you had at MIT.
Was satisfying. There are a lot of them, so I say we hard to pick out. If I want to pick out something about I think in general, I wouldn't be able to single out any single event. I think it's been very satisfying. I think one of the most satisfying things was seeing how a postdoc in the lab named David Krantz, for example, made a monoclonal antibody that was wonderful. We've made very few monoclonal antibodies in our lab compared to for a lab that's been around for a long time. But they've all been really very valuable and useful, and each one has been pretty significant. I will mention one other thing. Maybe this is not a good time to bring, not a very happy episode, and that had to do with that whole problem in Baltimore and Teresa Nishikari were accused of fraud, and became a very big political football in the arena because there were many people who jumped in and tried to take advantage of that situation. People were not at MIT, and I was caught in the middle of it.
I'm afraid I did that.
with the help of my friend here, Gene Brown. I was in
I asked him to take a look at the paper.
and for the next 10 years, I was caught up in that imbroglio, that including having to testify before Congress about this. So it was a pretty difficult situation. And it does bring up the whole matter about how we treat in science the distinction between error, fraud, mistakes, and so on. And how people who don't want to make those distinctions can blur the distinctions and create all kinds of problems. It was a pretty horrible experience all around.
Herman, as you look back on your career, is there anything you would like to have changed when you think about it?
I guess there's some papers I should have written sooner than I did. Are there any papers I should not have written? Yes. I'll mention one that was a pretty unhappy experience. By and large, I've had wonderful postdocs and no problems with any postdocs or graduate students. One postdoc, however, was a bit of a problem, and he did some work that looked okay, but then he pressured me to try to submit it to Nature. It was happily rejected. I was very happy to say, finally, because I subsequently found when I tried to repeat it that I couldn't repeat it. But then I saw the paper was published anyway. It was submitted for publication and had appeared without my approval. But
Without your name on it?
Huh?
Without your name on it?
No, my name was on it. And that, I think, was sort of around the time when many journals were saying they require every author to sign off and give approval for a paper being published. That was not the general practice, and it is now. And I would say that it should be, yeah.
Is there anything about how the department works?
How the department works?
At any level, administrative or teaching or research organization, or that looking back to say, we did it better then, or maybe we could learn something from the medical school experience that we haven't incorporated that might be helpful?
Well, in general, I really like and admire the MIT administration, and the way it's kind of structured so that faculty can have input if they want. But if they don't want, they don't have to get drawn into it a lot. And so there are people who want to do a lot of administrative work, and they can. And they do it. And I think the faculty can pretty much find its own level of activity to focus on what they want to focus on. And I think that's really how it should be. So I really think that the way things run at MIT, I'm pretty satisfied with and have been right along. Whereas at the medical school, I must say I was really involved in problems that I really had no interest in but was forced to. In fact, Washington U was sort of a very special school, I must say, in one respect. It was a school that was full-time. You know, most medical schools, including Harvard, people would practice and do teaching and do some research, had mixtures. At Washington University, when I went there, they had an exclusively full-time system. You just were paid by the university, had no outside income from anything else. And now it's different, of course. Now there's a lot of people on the faculty consult. They do a lot of, at MIT they do a lot of consulting. Some of them have companies, and it seems it's become increasingly accepted as a reasonable thing to do. But must say, I'm not entirely comfortable with that. Maybe it's old-fashioned. But I do remember once hearing Conant, who was a president of Harvard, having said that any professor at Harvard worth his salt can double the salary by consulting without his influencing or impacting on his ability to do academic work. That may be true of some people, but it was not true of me. I couldn't. I was involved in, since being on the board of some directors of some a company now defunct, and it took time and it took energy, and I found spending a lot of time at those things, it really draws one away from the lab, and I don't think so. For me, it was not a great activity on the side.
Did you find the quality of teaching in the medical school to be good when you were a medical student?
I think as a medical student, I learned more from my fellow students than I did from the faculty.
That's probably still the case.
I thought it was awful when I was a medical student. Amazingly awful.
Well, I still get feedback from students from here who go to Harvard Medical School. I still get feedback from those students.
I had a student
And
once.
most of it is not nice.
Yeah, right, I'm sure that's true. Yeah, I'm
One
sure.
particular problem, the idea is that you have God knows how many people teaching a course, because the idea is only an expert can give 3 lectures on something, and then you need somebody else for the next topic who's the expert.
It's that team teaching is really very.
That's very bad.
Yeah.
I had a student once here, a very good student, advisee in the Department of Pre-Med, and I advised her not to take immunology here. I said, you'll get it in medical school, take things here that you're not going to get in medical school. And she came back to visit about a year later, and I said, well, how's the immunology, or 2 years later? She said, oh, it was a terrible course. All these experts came in, and especially, she said, I more or less followed it, but we had people in the class that majored in history, and they didn't have a clue what was going on. All these world-famous people at Stanford in immunology, but they talked, gave them everybody the latest things, and the students, especially ones who weren't that well prepared for that, just totally lost. So I think that kind of teaching is really, I think the level of teaching here is far superior to what you get at medical school.
I'm not so sure that it's because the teachers are because it's a team teaching thing.
Mm-hmm.
I think what I've seen at MIT is a dedication to teaching.
Yeah,
and
that's
how
also
to teach.
true, yes.
At the medical schools,
Yeah.
there's not a dedication to teaching. You come in and you do your thing. But if there were more emphasis on how to communicate your ideas and how to simplify your slides and get to the bottom line, I think the quality.
But then you need to feel more responsibility for I think the trouble with team teaching also is not just there's different people, but nobody really identifies with making the course a real success because you're just giving 3 lectures on it.
Yeah, I totally agree. But it's not because it's team teaching
Yeah.
per se.
Another big difference in the graduate teaching. I mean, I was really struck from the beginning, and increasingly it's clear too. At MIT, graduate teaching graduate students, is taken very seriously. In medical school, there were some graduate students, but they essentially were, in the English system, kind of like apprentices. They worked in their mentor's lawyer, and that was it. They'd go to some seminars. There was very little coursework, very little thought about giving any formal teaching. Well, I think you're all exhausted.
You are.
Well, thank you very much.
You're welcome.